Hi guys, welcome to lesson 3 in our sort of introduction to the cardiovascular system, focusing upon the heart. Hi guys, welcome to our third lesson in this series on this introduction to the cardiovascular system, sort of focusing on the heart, but not entirely. The first thing I want to do, one of the first things I did in the very first lesson was I introduced you to a dilated cardiomyopathy case, which is a very good example of congestive heart failure, which is something we're going to focus on a lot during this block. I have a question, and if you want to, you can pause and look at it before I give you the answer. The patient had a soft holosystolic murmur. So, of the three choices below, which of these do you think is most likely causing the murmur? So, if you want to think about that, pause now. And now I'm going to give you the answer, and it is mitral insufficiency. So remember, insufficiency is a leaky valve. That means it's letting blood flow back through the valve when it's closed, meaning when blood should not be allowed to flow back through the valve. A stenosis is a narrowing of the valve, which takes place when you inject blood through an open valve that is narrower than usual. And because it's narrower, the velocity of flow increases, and it causes you to be able to hear a murmur. So how do we know? How do we know it was mitral insufficiency? Well, for insufficiency, we're looking for a closed valve. So in this case, we're looking for a valve that is closed during systole, okay? Or we're looking for a valve that is open during systole. So let's look. If we're talking about a stenosis, we're looking for a valve that's open during systole. If we're talking about an insufficiency, we're looking for a valve that's closed during systole. So let's take a look at our Wiggers diagram, which is one of the things I asked you guys to focus upon when you went through these videos or quickly read through the notes, one or the other or both. I asked you to actually study this. This is your Wiggers diagram, so let's take a look at ventricular systole. We have filling going on during diastole. Our blood volume is going up in the ventricle, and our pressure now is gradually rising as that ventricle sort of blows up like a balloon. And then this little hump is the atrial contraction. We know that because it corresponds with the P wave. And then the pressure in the ventricle, which is in the red, shoots up, and it goes above the left atrial pressure, and this is where your left AV valve closes, the mitral valve, or the tricuspid valve on the right. This is left, and you can tell that from the pressures, they're high. And so the mitral valve is closed at this point. Our ventricle is contracting, and we have begun ventricular systole. So we have a closed mitral valve and a closed tricuspid valve. So mitral insufficiency and tricuspid insufficiency are possibilities. Okay. On the other hand, here we have a point where the semilunar valve opens. That's during systole as well. So on the left side is the aortic valve, on the right side is the pulmonary valve. So during systole, the aortic valve on the left is open, the mitral valve is closed. So our possibilities for a systolic murmur are aortic stenosis or mitral insufficiency. Mitral insufficiency is a choice, aortic stenosis is not. So this was the one that you should have concluded was the correct answer. Okay, had it been a diastolic murmur during diastole, your mitral valve is open and you're filling the ventricle, your aortic valve is closed because the pressure in the aorta is high, the pressure in the ventricle is low, blood wants to flow back from the aorta to the ventricle and it can't. It's prevented by the valve. And so, so you're looking for aortic insufficiency or mitral stenosis during diastole. You're looking for aortic stenosis or mitral insufficiency during systole. Okay, and in this case, mitral insufficiency was the only one. So what you have is you have blood wanting to flow from high pressure to low pressure, from the ventricle to the atrium, but it's not allowed to do that during systole because that valve is closed. If the valve is insufficient, if it's leaky, then blood is going from high pressure to low pressure when it shouldn't be allowed to, and that is causing your murmur. You can hear that. Okay? All right. All right. Now, I emphasized to you guys that there were only certain things that I wanted you to concentrate upon and study, okay? And this video, this lesson, this third lesson in particular, is chock full of things that I just want you to be exposed to. Things where you can just play the video through, sit back and listen, look at the slides, and just digest. And if you don't completely understand everything I've said, don't sweat it. I just want you to have been exposed to it because things like the autonomic nervous system, which I'm going to bring back, which I know you're going to hear about. I can't keep people from, it's so integral to the function of the cardiovascular system, I can't keep people from talking about it. As a matter of fact, I'm going to talk about it before we actually do the autonomic nervous system lesson. Okay, so I just want you to be exposed to it. I want you to know what the sympathetic nervous system is, what the parasympathetic nervous system is, kind of what they do. Okay, not necessarily memorizing or remembering all the details yet. You will eventually, but not yet. I just want you to be exposed to it so you know what we're talking about when we bring it up before I get a chance to give you the details. All right? Okay. So this lesson in particular is chock full of stuff like that. So I'm warning you again right now, don't sweat it. You know, we're not trying, we are overloading you with information. We know that. That's part of being a medical student. But I don't want you to think it's any worse than it actually is because you're going through things like this and studying every detail. I don't want you to know every detail. We will cover the action potential in enormous detail in a lesson coming up. Okay, I don't need you to remember or even understand all the details yet. Okay, just want you to be exposed to the generalities. All right, having said that and emphasized it, let's go ahead and move on. So I said that, you know, we're talking about a systolic murmur. Ventricular systole is when your ventricular muscle is contracting, and of course that process of getting that muscle to contract starts with the propagation of the action potential. So here's our action potential, and again, you will notice that this action potential is prolonged, okay? Skeletal muscle or neural action potential will come right back down. This one is pretty long, and as a matter of fact, it's so long, it's almost the same duration as the contractile response, the generation of force and the relaxation itself. And because you can't fire another action potential until this one's over, the muscle is already most of the way relaxed before you can fire another action potential. And that means that you won't get a process that's called summation, which results in tetanus, which is something that you typically see or can see in skeletal muscle. You can't see that in cardiac muscle. You can't fire this action potential over and over again and get a bigger and bigger contractile response. It's going to be most of the way relaxed before you can fire another action potential. Okay? So the action potential is basically the same duration as the contractile response. Its purpose, really truthfully, in terms of cardiac muscle, is to open calcium channels. Okay, calcium channels in the cell membrane of the cardiac muscle, called the sarcolemma, are gated by voltage. They open when the voltage goes up. Okay, sodium channels are the same way. They open when voltage goes up. Okay, and that allows calcium to flow from high concentration to low concentration, from outside the cell to inside the cell, into the cytosol, through the open channel. Okay, and that causes a rise in cytosolic calcium, and that is what causes your contractile response. It causes your cardiac muscle to contract. Another thing that causes your cardiac muscle to contract is calcium release from the sarcoplasmic reticulum. The sarcoplasmic reticulum, or the SR, as we will typically call it, it's a very highly developed endoplasmic reticulum, for those of you who are familiar with the organelle and other cells that maybe aren't as excitable. Okay? It is an intracellular organelle, okay? And it has numerous calcium pumps that do active transport of calcium, and that causes calcium to be accumulated at high concentrations inside the SR. So calcium is transported from the cytosol, where it's low, to the lumen of the sarcoplasmic reticulum, where it's high, through the auspices of active transport through the SR calcium pump in that SR membrane. And it also has release channels called ryanodine receptors in it. Okay, and the calcium will be released from the SR during systole into the cytosol as it flows down its gradient from high concentration to low concentration to help cause the contractile response. It does that. It's helped along by the existence of T-tubules, or transverse tubules. T-tubules are nothing more than cell membrane that dips down into the inside of this cell. So one common mistake that medical students make is they treat this like an intracellular organelle, which it really isn't. The inside of this T-tubule, it dips in and comes back out, is just extracellular space. That's all it is. It's continuous with the extracellular space. And what it does is it brings calcium into the interior of this cell. And it really just brings the extracellular space into the interior of this cell. Okay? And that brings it close to the myofilaments, so the calcium doesn't have to diffuse very far. So T-tubules they're just cell membrane that dips down into the cell. They contain the voltage-gated calcium channels that are opened in response to an action potential, which will propagate down this membrane into the cell. And the calcium that comes in across this membrane binds to the SR calcium release channel, or the ryanodine receptor, and opens it, and that causes calcium release from the SR. So we call that calcium-induced calcium release. Calcium-induced, coming in from the outside of the cell through the voltage-gated calcium channels that are in the sarcolemmal membrane, calcium release from the SR caused by opening of SR calcium release channels in the SR membrane. Okay, and this is not this is relevant. It's medically relevant. You actually get decreased SR calcium concentration in chronic heart disease. And this can cause a decreased SR calcium release in heart failure. And as you will find out, if you get less calcium in the cytosol during a contraction, that means less force of contraction. That means your heart muscle isn't contracting as hard. And that means that your cardiac output will fall. Okay? All right. So calcium goes up during ventricular systole when cardiac muscle contraction is taking place, and we can see how this contraction sort of takes place. It takes place because of the existence of these myofilaments here. So we have our contractile proteins in cardiac muscle. We have tropomyosin, and we have actin here, and we have troponin, which actually has three different subunits, troponin T, I, and C here. Okay, so myosin forms your thick filament here, okay, and these form your thin filament. okay? And these two are going to interact to cause the cardiac muscle to contract in response to calcium. And these myofilaments are bundled up in what are called myofibrils, which run longitudinally through the cardiac muscle cell, okay? So you can see this bundle of fibers, and this is your cardiac cell, and it's running longitudinally through the cell, and you can see as contraction takes place, the two ends of the cell are brought together with some force. And that's because calcium binds to troponin C and causes these two types of filaments to interact to cause contraction, okay? So the higher the calcium concentration, the higher the force of contraction, and this is one of the things I did want you to remember. Now I've said it, now you remember it, okay? This is what positive inotropic drugs can do. So your calcium is coming across the sarcolemma through L-type calcium channels here. As the action potential propagates down your T-tubule, that causes calcium-induced calcium release, okay? So when that action potential ends, these SR calcium release channels close because the calcium channels, which are now closed because the voltage went down, they're not bringing calcium in across the sarcolemma anymore. Because there's no calcium coming in across the sarcolemma, the SR calcium release terminates because there's no more calcium-induced calcium release, okay? And calcium in the cytosol goes down, and that's because it is removed primarily by the SR calcium pump, which I described earlier, which is using ATP to pump calcium from the cytosol back into the SR, and primarily because of the sodium-calcium exchanger, which is using the sodium gradient. So sodium flows from high concentration to low concentration into the cytosol to pump calcium out of the cell from low concentration to high concentration. So this is secondary active transport, which was described to you in an earlier lesson. The sodium gradient is set up by the sodium pump, the sodium-potassium ATPase, which pumps sodium out and potassium in. And so the gradient that is set up by this is used by this to move calcium out, secondary active transport, okay? And because the calcium in the cytosol falls, the contraction stops and your muscle is allowed to relax as the ventricle fills and stretches those fibers back out, okay? There you go. All right, so that is the end of our first video in this lesson. In our second video, we will introduce the autonomic nervous system and what it does to all of this. Don't panic. If you don't understand all of it, we'll get to it, okay? And then after that, we'll talk about how positive inotropic drugs work. Just as an example of how cardiac muscle can function and how all this stuff can come together, don't panic. I don't expect you to remember the details. All right? Thank you for your attention.